Hello and good day, everybody. We are Group 14 from the course SIX1015 Occurrence 1, and today we are going to present to you about the topic CRISPR in agriculture field. My name is Ishrada Hanim binti Muhammad Jamaluddin, and here are my group members.
My name is Kelvin.
My name is Justin Titi.
I'm Rashim Pritkaur.
I'm Aisha Hana.
I'm Shashwini. Okay, so next we'll move on to the definition of CRISPR. In its original form, CRISPR or Cas9 is a homing device, which is a CRISPR component that directs the Cas9 enzyme, a restriction endonuclease enzyme, to a target segment of DNA. They function together like a genetic engineering cruise missile, disabling or repairing genes or inserting something new where the Cas9 scissors have made cuts. CRISPR is also known as a segment of genetic material found in the genomes of prokaryotes, such as some bacteria and archaea, that consist of repeated short nucleotide sequences interspersed at regular intervals between unique nucleotide sequences derived from the DNA of pathogens, such as viruses that have previously infected the bacteria and that protects the bacteria against future infection by some pathogens. Next, we'll move on to the mechanism of CRISPR. In a document, if we suspect we misspelled the word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR in short form is clustered regularly interspaced short palindromic repeat. CRISPR consists of two components, the Cas9 protein that can cut DNA and guide RNA that can recognize the sequence of DNA to be added in. They use CRISPR-Cas9. Scientists first identified the sequence of human genome that's causing health problem. Then they created specific guide RNA to recognize that the particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then the complex is introduced to the target cells. It locates the target letter sequence and acts as the DNA. At that part, scientists can then edit the existing genome and either modify, delete, or insert new sequences effectively, making CRISPR-Cas9 as a cut and paste tool for DNA editing. Next, we'll move on to the history of CRISPR-Cas9.
Thank you, Shah. So we are going to explain the history of CRISPR. So the first encounter with CRISPR is in the mid-80s during the study of isozyme conversion of alkaline phosphate. One of the researchers noticed that the same sequence of the DNA fragment appeared numerous times in various E. coli clones. Next. Okay, the method they know at the time made it impossible for them to read the repeating sequence, and it took them several months to precisely read the CRISPR region sequence. The characteristic of the repeating sequence was so surprising and unexpected in the study that it was noted in the discussion even though its significance was unknown. Next. Okay. Soon, an identical sequence also discovered in other E. coli strains, Shigella dysenteria and S. enterica sevorva typhimurium. Mojica and his colleague made a significant breakthrough when they discovered identical repetitive sequence in the Halopherax mediterraneae while researching regulatory mechanisms that allow exceptional halophilic archaea to adapt to high cell environment. Next. According to the findings, this repetitive sequence may be important in the regulation of gene expression. The same researchers also discovered a similar repeated sequence in Halopherax volcani and hypothesized that it was involved in replicant partitioning. As DNA sequencing technology advanced in the 90s, scientists had access to whole genome sequences. They discovered the unique repetitive sequences in a variety of bacteria and archaea genomes and gave them various names, including short regularly spaced repeats, spacer interspersed direct repeats (SPIDR), and large cluster of tandem repeats (LCTR). In 2002, Jensen and her colleague coined the term CRISPR, or clustered regularly interspaced short palindromic, and it became widely recognized by the community working on these sequences. So I will pass the next section to the other members. Thank you.
So moving on to the next part of the presentation, which is the benefits of this CRISPR-Cas9 technology. Okay, so these are some of the known benefits of CRISPR technology in agriculture field. First of all, CRISPR technology helps to produce better crops with desired traits. The genome of the plants can be altered thanks to this technology to meet the farmer's need. The genetically modified plants will be resistant to pests, disease, and harsh weathers. This indeed will help the farmers to increase their population yield without worrying about their crops anymore. Moving on to the second benefit, which is the crops have increased nutritional values. We as a human requires nutrition for our growth, energy, and for healthy life. Thus, it is very beneficial for us to consume these foods with enhanced nutritional values, as we can obtain the nutrients we need faster. Here are a few examples of plants with increased nutritional values. Genetically modified potatoes contain lower acrylamide level. Genetically modified corns have better starch quality, while genetically modified wheat contains high fiber level. Last but not least, CRISPR technology reduces food wastage. Fruits are known to have this browning effect, which will make the fruit turn bad. The browning effect is a process of food turning brown due to chemical reaction, usually enzymes that take place within the fruit. The negative impact of this browning effect is that it affects the color, flavors, texture, and nutritional values of fruit. This makes the fruit looks ugly. Thus, customers will lose interest and don't want to buy the fruit. When the fruit is left to rot, this is where the wastage of food occurs, indirectly causing huge economic loss to our country. Luckily, CRISPR technology can help us to solve this problem. These are the examples of browning effect featured by some fruits. Okay, next, we'll move on to the current state.
Now I will be explaining about the current state of science and technology development. CRISPR became the most widespread technique to improve monocot and dicot crops for a variety of traits including yield, quality, disease resistance, and climatic variation resistance. For example, the genome of cereal crops and fruits have been edited using CRISPR technology. Next, CRISPR is used to improve the yield and quality. CRISPR aims to create the largest milestone in genome editing in the modern technology by developing cultivars with strong nutritional values and resistance characteristics. Furthermore, CRISPR is frequently used to develop disease-resistant crops. The main cause of disease, which result in huge reduction in crop yield and viruses, fungi, nematodes, insects, and bacteria. CRISPR has also been recently used to edit glutenine genes to develop celiac-safe cultivars. These methods produce offspring with silenced and deleted guidance, which may reduce exposure to CD epitopes. Lastly, by developing climate-smart or abiotic stress-resistant crops, the plant breeding system has been modernized. According to studies, CRISPR method holds a great potential for developing climate-adapted crops. CRISPR could also alter genes to facilitate resistance against many abiotic stresses such as drought, high temperatures, heavy metals, as well as nutrient deficiencies. That is all for my part. Next, Aisha will be explaining about the examples in sustainable development.
Okay, there are a few examples on how CRISPR is a sustainable development technology, with the first one being CRISPR can reduce greenhouse gases emission. This is because gene editing can enable crop plants to grow bigger and deeper roots that are incredibly effective in capturing carbon and keeping it sequestered in the soil for longer periods of time instead of releasing it to the environment. These gene-edited plants could reduce up to 46% of the excess carbon emitted annually worldwide if they became a fixture in the agricultural food chain. Next, CRISPR could also help in reducing pollutions. Genome editing is also used to improve the retention of nutrients in plants and their ability to take up nutrients from the soil. This reduces the reliance of unsynthesized fertilizers, cutting the pollution emitted in the manufacturing process, as well as the waste from the fertilizers that flow through the river that could promote algae growth, which we know as eutrophication. Next, I'm going to pass the floor to the next presenter.
Thank you, Isha. Next, we'll move on to the challenges in implementing. CRISPR-based therapies are rapidly advancing with applications involving cell replacement therapies, gene drives, and diagnostics. The use of CRISPR, both ex vivo and in vivo, is hindered by the immunogenicity of CRISPR components, limited delivery, low specificity of target effects, DNA damage, and repair pathways. CRISPR limitations are being addressed through advances in controllers, such as degraders or inhibitors of Cas9, conjugation strategies to endo Cas9 with new functions, and base and primer editors that do not produce double-stranded breaks. The application of CRISPR-Cas9 in the germline is considered more problematic because of the risk of causing various mutations and side effects and transferring undesirable changes to the future generations. So next, we'll move on to the recommendations presented by Aisha Hana.
Thank you, Shah. So I will be talking about the last point of the presentation, which is recommendations of CRISPR application. So what are the things that we can do in our country regarding CRISPR? CRISPR can be applied in Malaysia's agriculture sector through several ways. The first one is by educating the public about the importance of CRISPR and how they function, as well as giving benefits to the others. For example, the government or the association in charge may launch events, exhibitions, or even advertise on mass media for the public to acknowledge them. Next, the government should increase the funding in the agriculture field so that it is easier for the involving parties to continue the activities, as there would be no shortage of financial support. Furthermore, scientists can be hired to invent some sort of seeds that may produce high-quality products, and the seeds can be distributed to the agriculture society. So we have come to an end of our presentation. That's all from our group. Thanks for listening.